Welcome to this lecture on the biochemistry of metabolic processes. Today, we are going to explore a fundamental and incredibly elegant process called transamination. If you have ever wondered how your body manages its protein building blocks, extracts energy from them, and safely disposes of toxic waste, you are in the right place. Let us dive in and uncover how your cells perform these molecular trades every single second to keep you alive and healthy. Now that we have introduced the topic, let us start with a simple analogy to build our intuition. Imagine you are running a massive, bustling restaurant kitchen. Suddenly, you realize you have way too many hamburger buns, but absolutely no hot dog buns. Instead of throwing the hamburger buns in the trash, you find a neighboring restaurant that has the exact opposite problem. You simply trade your surplus for what you need. This simple act of swapping is exactly how your body handles its resources at a microscopic level. It is a highly efficient system of molecular trading. Building on that restaurant analogy, your cells face a very similar problem with protein building blocks called amino acids. In the incredible factory of your body, you sometimes have too much of one specific amino acid and not enough of another. To solve this, your body uses a brilliant swapping mechanism called transamination. It is a molecular trade where one molecule hands over its defining feature to another. By doing this, it instantly transforms both molecules into exactly what the body needs at that specific moment, without wasting any valuable resources. With that intuitive understanding in mind, let us look at the formal biochemical definition. Transamination is a specific chemical reaction where an amino group, which contains nitrogen, is transferred from an alpha amino acid to an alpha keto acid. An alpha amino acid is simply a carbon skeleton holding onto a nitrogen group, while an alpha keto acid is a similar skeleton with an oxygen double bond instead. When this reaction occurs, the original amino acid loses its nitrogen and becomes a keto acid. Simultaneously, the original keto acid gains that nitrogen and becomes a brand new amino acid. Knowing what the reaction does naturally leads us to ask how it happens so quickly. This elegant swap does not just happen on its own. It is catalyzed by a special class of enzymes known as aminotransferases, or transaminases. Furthermore, these enzymes cannot work alone. They absolutely require a helper molecule called pyridoxal phosphate, often abbreviated as PLP. You can think of PLP as a temporary holding carrier. It grabs the amino group from the first molecule, holds onto it for a split second, and then safely hands it over to the receiving molecule. Now that we know the mechanics, why does this molecular swapping matter so much to your daily survival? First, transamination is the critical first step in breaking down the proteins you eat into usable energy. Because the human body cannot store excess nitrogen, we must safely strip the nitrogen off amino acids before we can burn their carbon skeletons for fuel. Second, this process allows your liver to manufacture non-essential amino acids from scratch. This means you do not have to rely entirely on your diet for every single building block your body needs. Beyond just energy, this process is an absolute matter of life and death. If this transamination process were to fail, toxic nitrogen would rapidly build up in your bloodstream as ammonia. This accumulation would lead to severe brain damage or even death. Because these enzymes are so vital, they also serve as important medical markers. Doctors routinely measure the levels of these transaminase enzymes in your blood during checkups. Elevated levels can help detect liver damage early, making this concept vital for both basic biology and clinical medicine. Having discussed the biological importance, let us look at the mathematical and chemical representation of this process. Conceptually, this formula shows the exact balance of the trade. A1 represents the first amino acid, which is the starting molecule holding the nitrogen. K1 represents the first keto acid, acting as the empty carbon skeleton waiting to receive. When the reaction moves forward, the amino group is transferred, creating K2, the new keto acid, and A2, the brand new amino acid. The double arrows indicate this reaction is completely reversible. If the concentration of the starting materials increases, the reaction pushes forward. Conversely, if the products build up too much, it flows backward to restore chemical balance. Let us apply this formula immediately with a concrete example. Suppose your muscle cells are working hard and generating a large amount of the amino acid alanine. The body must transport this nitrogen safely to the liver. We start by identifying alanine as our primary nitrogen donor and alpha-ketoglutarate as our empty acceptor. We select the specific enzyme, alanine aminotransferase, or ALT, to catalyze the swap. We set up our equation with ALA and AKG on the left. During the reaction, alanine gives up its nitrogen to become pyruvate, which the cell uses for energy. Simultaneously, alpha-ketoglutarate accepts the nitrogen to become glutamate, safely trapping the toxic nitrogen for transport. Our final answer shows this elegant solution. Following our first example, let us look at a second crucial reaction. Imagine the liver is now preparing to excrete the excess nitrogen through the urea cycle, which absolutely requires the amino acid aspartate. The liver currently has abundant glutamate, carrying nitrogen, and empty oxaloacetate. We identify glutamate as our donor and oxaloacetate as our acceptor. This time, we use the enzyme aspartate aminotransferase, or AST. We set up the equation with GLU and OAA on the left. Glutamate gives away its amino group, reverting to alpha-ketoglutarate. Oxaloacetate receives the nitrogen, instantly converting into our target, aspartate. Our final answer shows the newly formed aspartate, which can now directly enter the urea cycle to safely flush nitrogen from the body. Now that we have calculated these specific examples, let us visualize the actual molecular mechanism on screen. On the left side of the diagram, you can see the starting amino acid, carrying its nitrogen group, highlighted as NH3+. Below it is the starting alpha-keto acid, which has a double-bonded oxygen instead of nitrogen. Notice the reversible arrows in the center, driven by the aminotransferase enzyme and its PLP helper. As we move to the right side, you can clearly see the result of the swap. The first molecule has lost its nitrogen and gained an oxygen, becoming the new keto acid. The second molecule has accepted the nitrogen, transforming into the new amino acid. This perfectly illustrates the elegant trade we just performed. To bring everything together, let us review the most important ideas we have covered. Transamination is the body's elegant and highly efficient molecular trading system for managing nitrogen. By swapping amino groups, your cells can seamlessly balance their inventory of biological building blocks. Remember that this process is entirely reversible, strictly requires the helper molecule PLP, and is driven by specific enzymes like ALT and AST. Mastering this foundational concept is the absolute key to understanding how our bodies extract energy from proteins and safely dispose of toxic waste. You have now covered the full topic and understand a vital mechanism of human survival.